Hello, and welcome to our last little part of our unit on the cell. We just wrapped up the part of the cell unit that had to do with transformation of energy. So we looked at cellular respiration, we looked at photosynthesis and the way that organisms gather, transform, and use energy. And in these last couple of chapters, we're going to deal with this other important life function of transmission of information. So in this chapter, we're going to deal with cell communication. But remember that we can think about transmitting information both through space and time. So this time we're going to think about transmitting information through space between different cells in an organism or between different unicellular organisms. And then in the next web lecture on the cell cycle and mitosis, we're going to think about transmitting information through time from one generation to the next. So remember that this idea of transmitting information through space is going to be very important for maintaining an internal environment that's different from the external conditions, otherwise known as homeostasis. So information has to be brought into the organism and received about the local conditions to be able to respond to those conditions to maintain that internal environment. And then, of course, responding is the other big component of this, responding to other organisms, responding to factors in the environment to be able to maintain that homeostasis. So let's think about cell-cell interactions. So unicellular organisms must contend with constant shifts in its environmental conditions in terms of temperature, salinity, acidity, pH. All of these things need to be monitored and regulated. And then, of course, cells and multicellular organisms must communicate and cooperate with each other to produce coordinated responses. So this is the example given in your book of the impala being chased by the cheetah. This has to be a whole body, multiple system response that all happens in a very coordinated way to be able to escape from this cheetah. So cells must obtain information and respond to it, and the cells must interact with other cells to produce this kind of coordinated response. So cells can signal to each other. They need to interpret the signals that they receive from other cells and from the environment. And so these signals are most often in the form of chemicals. So for example, the signal that's coordinating this response on the part of the impala is a molecule called epinephrine, also known as adrenaline, that is creating that multiple system, organism-wide fight or flight response that's going to hopefully save this impala's life. So how are these external signals converted to a response within the cell? So communication among microorganisms provides some insight into how cells send, receive, and respond to signals. So the example in your book is the two mating types in the yeast cell Saccharomyces cerevisiae, and it has two mating types, A and alpha. And so the cells of these different mating types can locate each other by secreting these signaling molecules that are going to be received by only the cell of the opposite type. Here we see that the alpha cell secretes this alpha factor. The A cell has receptors for alpha factor, but not for A factor. Okay, so if another cell that was A type was around sending these signals, the signals would not be received because this A cell does not have the right kind of receptors. Similarly, the signals sent out by the A cell are going to be received by receptors on the alpha cell that are specific to those A signals, and the alpha cell could not receive signals from another alpha cell because it does not have any receivers for those alpha factors. They receive these mating signals. They can only respond to them if they have the correct receptors on the outside of the cell. And then if they do receive that signal, if those mating factors bind to the receptors, it initiates a whole sequence of events that's going to include moving toward the source of that signal, the other cell, fusing membranes, and merging together to form an A alpha cell. So the binding of that mating factor at the cell surface is going to initiate a series of internal steps, actions on the part of the receiving cell that's going to be some kind of response to that signal. So this is called a signal transduction pathway, the series of steps that leads from the receipt of a signal to a response. So how did these cell signaling systems evolve? So we know that the same small set of cell signaling mechanisms shows up in many diverse species and many diverse processes. So the more you see something common to many, many different kinds of organisms that last shared a common ancestor a very, very, very long time ago, you can infer that those mechanisms evolved somewhere in the common ancestor of all of those groups that possess it. So we can say that some of these signaling pathways are extremely old, dating back to the very earliest forms of life. And the molecular details of signal transduction in yeasts and mammals are very similar. So this indicates that these pathways are also very, very old. So ancestral signaling molecules likely evolved in prokaryotes, since prokaryotes evolved before eukaryotes. And those were passed down and adopted for use in their multicellular descendants. So we know that cell signaling is critical among prokaryotes, among eukaryotes, among pretty much all organisms. So let's take a look specifically at signaling between unicellular organisms. So unicellular organisms live together. There are going to be other organisms in their environment, and they're going to communicate with one another. And so often this communication, what they're talking about is changes in the environment so that they can all kind of coordinate their responses to these changes in the environment. So signaling pathways that respond to population density are called quorum sensing. So basically, they're going to communicate in such a way that they can detect how many individuals there are in an area, how dense their population is locally. So they're going to release species-specific signaling molecules, and they're going to do this when their numbers reach a specific threshold. So that means they need to detect how many other members of the same species are close by, and then they're going to send out signals that create particular behaviors in the surrounding bacteria. So the response to the signal molecules is going to vary across species. So examples of responses to quorum sensing, some bacteria use it to help glue a community of microbes to form a biofilm. One example of a biofilm is the plaque that forms on your teeth when bacteria reach a certain density. Also, the film that forms on stagnant ponds, so we might call this pond scum, is an example of a biofilm that is created when there's a certain density of bacteria. When they receive that signal, they all congregate together to form this film, and that film is going to have some of the benefits of a multicellular organism. So bacteria and light organs of the bobtail squid are going to secrete enzymes that catalyze a light-producing reaction, but only when they reach a particular density. And then an organism called a slime mold, when its density becomes too high, those individual single cells are going to aggregate into this multicellular slug-like body, and that slug-like body is actually going to migrate away to reduce the density of those unicellular organisms. And then when they get to an area where the density is low, they have a lower population in that area, then they're going to disaggregate again into their unicellular form. So when we get to multicellular organisms, now we have a situation where you might want to signal to cells that are in your immediate area, but you also might need to signal to cells that are a very long distance away from the origin of that signal. And so in multicellular organisms, we have both local and long distance forms of signaling. So short distance signaling can be limited to just very nearby neighboring cells. And so in this paracrine secretion, cells might just release particular molecules that are just going to diffuse away and be received by any nearby cells that have an appropriate receptor for them. But when we're talking about long-distance signaling, we have a couple of different systems that can be used. One is the nervous system, which I'm not going to discuss in very much detail here. We're going to have a whole chapter on the nervous system later on in the semester, but this allows a signal to be transmitted from a very long distance to a very specific region in the body. So it's going to be very specific, and it's going to work over long distances. The one that we are going to talk about in more detail today is the endocrine signaling. And this is signaling by molecules called hormones that are going to be released into the blood, usually by glands. And because they're carried in the bloodstream of animals or through the vessel system of plants, it's going to be delivered pretty much throughout the multicellular organism. So it's going to be fairly nonspecific. It's going to go everywhere in the body. The only cells that will respond to them are cells that have the specific receptors. So let's first think about local signaling. So cells in a multicellular organism communicate, just as we saw in single-celled organisms, through these signaling molecules. In local signaling, animal cells may communicate by direct contact. So we've seen this in animal cells in the form of gap junctions, in plant cells in the form of plasmodesmata. And in this case, these cell junctions are going to directly connect the cytoplasm of adjacent cells. And so signaling substances that are released by one cell can pass freely into adjacent cells, making this transfer of information very easy. And so local signaling in plants is not very well understood beyond this kind of direct cell-to-cell communication through plasmodesmata. But in animal cells, we know of a couple of other mechanisms for local signaling. And one is the one that I just mentioned a couple of slides ago, paracrine signaling. So animal cells can communicate using secreted messenger molecules that travel just short distances. They're basically just going to diffuse away from the cell that's secreting them. So these can include signals such as growth factors, which stimulate nearby target cells to grow and divide. And this is one class of these local regulators that are just going to move very short distances from the cell of origin. And this is called paracrine signaling. Another form of local signaling that we see in animals is associated with the nervous system primarily, and this is called synaptic signaling. If we look at a nerve cell coming down here, and the very end of the nerve cell comes into communication with its target cell through this tiny little space. This little gap is called a synapse. The signaling cells, which are known as neurotransmitters in the nervous system, are going to diffuse across that synapse and be received by receptors in the target cell. So this is going to be in response to an electrical signal. So if you think back to when we were talking about the sodium-potassium pump, we were talking about changes in the voltage across the membrane. So those changes in voltage are going to be what triggers the release of these signals in the synapse. If we think about long-distance signaling, both plants and animals use chemicals called hormones. So hormonal signaling in animals is called endocrine signaling, in which specialized cells in some kind of gland release hormones which travel to target cells via the circulatory system. So they go out in the blood everywhere, but the only cells that are going to respond to them are the ones that have the particular receptors for that signal. The ability of a cell to respond to a signal depends on whether or not it has those receptors. So it can be a fairly specific response based on the particular kind of receptors that are possessed by the target cell. Cell signaling happens in 3 stages. This was discovered by Earl W. Sutherland, who you read about in your book, and his colleagues, and they discovered how the hormone epinephrine acts on cells, and some of these experiments are described in your book. I encourage you to take a look at them. It's a very interesting story. But what he discovered is that cells receiving signals went through 3 processes. So for example, you couldn't just inject the epinephrine into the inside of the cell and expect the correct response. It has to go through an entire pathway, and that pathway is going to include reception. So the signal is received somewhere in the cell, either outside or inside. Transduction, which means that the signal is going to be relayed through intermediates of various kinds. This can often involve the amplification of that signal. And then a cellular response. What is the cell going to do as a result of receiving this signal? So in reception, the target cell detects a signaling molecule that binds to a receptor protein on the cell surface. So this is going to be that lock and key mechanism. The signaling molecule will only bind to the receptor if it is the right size, shape, polarity, has all the right features to be compatible with that receptor cell on the cell surface. So in transduction, The binding of the signaling molecule alters that receptor in some way and initiates a signal transduction pathway. So this is often going to occur in a series of steps. So relay molecules 1, 2, 3. We'll look at this in more detail as we go. And then finally, in the response, the transduced signal is going to be in a form that can actually trigger the specific response that is desired in the target cell. So now let's focus on this first step, reception. So a signaling molecule is going to bind to a receptor protein and cause it to change shape. So the binding between a signal molecule, which we call a ligand, the term ligand implies this specificity that it has with the receptor. So the ligand and the receptor, and it's going to be highly specific. So that binding, remember when things bind with proteins, proteins tend to change shape. This is a theme that we've seen over and over again. And that change in the receptor shape is often the initial transduction of the signal. It's going to cause something to happen inside of the cell. So most of these signal receptors are going to be plasma membrane proteins. So transmembrane proteins. Signal will bind outside the cell, cause a response inside the cell. And these signal receptors are dynamic. So the number of receptors in a cell may change depending on circumstances, and we can use what we've learned already about how the cell works to understand this. If you need to upregulate receptors in a cell, you transcribe the DNA for a bunch more of that receptor protein that's going to go through that whole endomembrane system, ending up in a vesicle that's just going to be added to the cell membrane. So if that is sounding obscure to you, please go back and review that process. But basically, these chunks of the membrane with receptors in them can be added, they can be taken up, increasing or decreasing the number of receptors as the need arises in a cell. So the ability of a receptor to bind a hormone may change. Depending on the environmental conditions, a receptor may have more or less affinity for its ligand. And the sensitivity of a cell to a particular hormone may change over time, and this is mostly going to be a function of this number of receptors in the cells. And signal receptors can be blocked. We can have this competitive inhibition of signal receptors, and many pharmaceuticals and drugs and poisons work in this way by blocking these signal receptors. So these signaling molecules may be either hydrophobic, so nonpolar molecules, or hydrophilic polar molecules. They can also vary greatly in size. So hydrophobic signals diffuse right across the plasma membrane if they're small enough. And so in this case, the receptors are going to be within the target cell's cytoplasm. So the receptors are going to be located inside the cell. Hydrophilic signals, so these polar molecules and also large molecules, do not cross the plasma membrane. And these water-soluble signal molecules are going to bind to specific sites on receptor proteins that span the plasma membrane. They're never going to enter the cell. They're going to transmit their signal by changes in those receptor proteins. There are three main types of membrane receptors that we're going to be discussing. So one is the G protein coupled receptors, also known as GPCR. The second is receptor tyrosine kinases. We're going to mostly call those RTKs, and ion channel receptors. So G protein coupled receptors, GPCRs, are cell surface transmembrane receptors that work with the help of something called a G protein. So they're called G proteins because they bind a molecule called GTP, which is guanosine triphosphate. It's very closely related to adenosine triphosphate, ATP, and all of these G proteins are pretty similar in structure and function in pretty much the same way. So let's take a look at how they work. So here is the inactive situation in box 1. There's no ligand bonded to the receptor. The G protein is in its inactive form, and it has a molecule of GDP, guanosine diphosphate, attached to it. And there's an inactive enzyme somewhere else in the plasma membrane, or somewhere else in the cytoplasm. When that signaling molecule, that ligand, is received, the receptor becomes activated. This G protein is going to bind with the activated receptor. It's going to gain an affinity for that receptor, and this is going to be an enzyme that removes that GDP and replaces it with a GTP. So the GDP is going to be bumped off and replaced with a GTP, and now this G protein is activated Now it's going to have an affinity for this inactive enzyme. So when the activated G protein with GTP bonded to it interacts with this now activated enzyme, there's going to be some sort of cellular response. When the G protein dissociates from the enzyme, it's going to split into inorganic phosphate and GDP, and ready to repeat this cycle. So G protein coupled receptors are the largest family of cell surface receptors. You're going to see them over and over again in this chapter. And GPCR systems are extremely widespread and diverse in their functions. So now let's take a look at receptor tyrosine kinases, RTKs. So they are membrane receptors that transfer a phosphate group from ATP to another protein, and that is actually the general definition of a protein kinase. It's going to transfer phosphate groups to some protein. Let's take a look at how this works. These receptor tyrosine kinase proteins work in pairs, sometimes in small clusters, but that's an ongoing area of research. We're going to think of these as pairs of these receptors. So when both of these receptors have a signaling molecule attached to them, they're going to come together to form a single unit called a dimer. So each of these subunits has a number of tyrosine residues attached to them. In this case, we see 3 on each of them. So once these two subunits dimerize, come together, that's going to initiate the process of phosphorylation of each of these tyrosines. So 1 ATP for each tyrosine, and each of these tyrosine is going to be phosphorylated. So each of them has now a phosphate group attached. So this is the phosphorylated dimer form of this activated receptor. So because there are many of these tyrosine residues that can be phosphorylated, they can actually trigger multiple signal transduction pathways at once. So here we see 2 different relay proteins that are going to attach at different places that are going to end up creating 2 different cellular responses. So these are very versatile, and they're very good for creating these coordinated responses that incorporate different pathways all at the same time. So abnormal functioning of RTKs is associated with many types of cancers. So now let's look at this third category of receptors, the ligand-gated ion channels. So these receptors act as a gate that opens and closes when that ligand binds. So here we're seeing an example of a synapse, what we looked at before with that synaptic signaling. We've got the neurotransmitters, this is acetylcholine in this case, and then when acetylcholine binds with the binding site on this ligand-gated channel, that's going to cause a change in shape of this channel protein that's going to allow sodium ions to flow into this cell. So it's going to open and close when that receptor changes shape in response to the binding of the ligand. So when a signal molecule binds as a ligand to the receptor, the gate allows specific ions such as sodium or calcium through a channel in the receptor. So it's going to be very specific to the molecule that it's going to allow to flow through. So intracellular receptor proteins are found within the cytoplasm or the nucleus of target cells. So these small or hydrophobic chemical messengers are going to cross right across the plasma membrane. They're going to diffuse across, and they're going to activate these receptors. So let's take a look at what this looks like. So this is our small nonpolar molecule that's going to diffuse right through the membrane, and it's going to find a receptor within the cytoplasm, within the cytosol. So examples of some of these hydrophobic messengers are things like the steroid hormone that you see here, or also thyroid hormones of animals. And when the signal binds to the receptor, it forms a complex with it. So it's going to be an activated hormone receptor complex that in many cases will do something like pass into the nucleus and act as a transcription factor, turning on or off specific genes, but it's going to be an action within the cell that will only happen when this receptor is activated by the signal. So now let's look at the second phase, transduction. So what we're going to see are cascades of molecular interactions that are going to relay the signals from the receptors to target molecules in the cell. So this is kind of the intermediary between the receipt of the signal and the response. So we're going to see that cell signaling is usually a multi-step process, and that in the process of this multi-step pathway, there's an opportunity to greatly amplify a signal. So if a single molecule early in the pathway can go and activate a whole bunch of other molecules, and each of those are going to activate a bunch of other molecules, you can have a single signaling molecule producing many, many, many times more response molecules. And we'll also see that these multi-step pathways provide opportunities for very, very, very precise regulation and coordination of the cellular response. So again, this idea of homeostasis, keeping conditions within a very narrow set of optimal conditions, requires very fine-tuned coordination and regulation. So one of the changes that can be made through this whole transduction process is the phosphorylation of protein, the adding of phosphate groups to a protein. And this is a very common method of signaling. So this is going to activate proteins to do particular things. So phosphorylation and dephosphorylation of proteins is a widespread cellular mechanism for regulating protein activity. Protein kinases are proteins that transfer phosphates from ATP to another protein. And this process is called phosphorylation. So many of the relay proteins in these pathways are going to be protein kinases. So many relay molecules in signal transduction pathways are protein kinases, and this is going to create a phosphorylation cascade in which one protein kinase phosphorylates another protein kinase, activating it. That one is going to phosphorylate another protein kinase, activating that in this cascading action, this chain reaction of transduction. Then we need to somehow reverse this process to be able to shut this off, and this is done by protein phosphatases, which are going to remove the phosphates from these proteins in a process called dephosphorylation, so resetting these proteins to their unphosphorylated state. So this phosphorylation and dephosphorylation system acts as a molecular switch, turning activities on and off or up or down as required, ramping it up by phosphorylating, ramping it down by dephosphorylating as needed. Here's a diagram of how that works. So here we have the signaling molecule that's going to activate the receptor, which is going to activate a relay molecule. This activated relay molecule is going to interact with an inactive protein kinase number 1 to become an active protein kinase number 1. This active protein kinase number 1 is going to interact with this inactive protein kinase 2, phosphorylating it to become activated. So this is now an active protein kinase 2 with a phosphate group attached. Eventually, the phosphate group is going to be removed by a protein phosphatase. Here we have the final inactive protein in this pathway that's going to be phosphorylated by the active protein kinase 2 to become an active protein that's phosphorylated, resulting in the cellular response. Eventually, this one too is going to be dephosphorylated back to its inactive state. So this constant shifting between active and inactive states is what allows these signals to be turned on and off as needed. So many of these signaling pathways involve molecules called second messengers. So they're called second messengers because we think of the original signaling molecule that bound outside the cell as being the first messenger, so the primary messengers. And so these transduction molecules we call secondary messengers. There may be a series of secondary messengers. We call them all secondary messengers, all of the ones that are intermediaries in these pathways. So these are small, non-protein, water-soluble molecules or ions that can spread quickly throughout the cell by diffusion. So second messengers participate in pathways initiated by both GPCRs and also RTKs. So this is a common pathway to both of these kinds of receptors. And the most common of these second messengers are molecules called cyclic AMP and also calcium ions. So cyclic AMP, also known as CAMP, is one of the most widely used second messengers. You'll see it over and over and over again throughout your study of biology. And an enzyme called adenylyl cyclase in the plasma membrane is responsible for converting ATP, so that's regular ATP, adenosine triphosphate, into cyclic AMP in response to some extracellular signal. So let's take a look at how this transduction pathway works using CAMP. So first we've got our signaling molecule binding to our G protein coupled receptor, our GPCR. Next, this GPCR is going to attach GTP to a G protein. That activated G protein binds to adenylyl cyclase. So remember, this is the enzyme that's going to convert ATP to cyclic AMP. So now this is working as a second messenger. And what is it going to do? It's going to activate a protein kinase, usually something called protein kinase A, which is then going to produce the cellular response. So this whole pathway can be regulated very easily by activating or inhibiting this adenylyl cyclase. If you can inhibit the adenylyl cyclase, it's going to not create cyclic AMP and this whole pathway will not occur. So let's look at our other very common second messenger, calcium ions, and they're going to work together with a molecule called inositol triphosphate. We're going to call this IP3. So calcium ions, again, are used very widely as a second messenger, and calcium can function as a second messenger because its concentration in the cytosol is normally much lower than the concentration outside the cell. And this is because there are active pumps that pump calcium ions both out of the cell into the extracellular fluid, but also pumps that pump calcium into the endoplasmic reticulum, sequestering it there so that it can be used in circumstances like this as a second messenger. So a very low concentration inside the cytoplasm, so that a small change in the number of these calcium ions can represent a large percentage change in calcium concentration. This can be used very effectively, in this case, as a signaling molecule. So a signal relayed by a signal transduction pathway may trigger an increase in calcium in the cytosol. So pathways leading to the release of calcium involve IP3 and diacylglycerol, also known as DAG, as additional second messengers. Remember, we call all of them second messengers, even if one of them happens before the other one. So these two are produced by the cleavage of certain phospholipids in the plasma membrane, and we'll see how that works right here. So this is the generalized transduction pathway using calcium ions and IP3. So again, we have an example with the GPCR that's going to activate our G protein by adding a GTP to it. And this GTP is going to activate an enzyme called phospholipase C. And phospholipase C is going to cleave a lipid molecule called PIP2 into our diacylglycerol and IP3. The IP3 in this case is going to be the second messenger. So what the IP3 is going to do is it's going to come and bind to a ligand-gated calcium channel in the endoplasmic reticulum. Okay, so the binding of IP3 to the receptor on this channel protein is going to cause a change in shape in the protein that's going to open up this channel and let calcium ions flow from the endoplasmic reticulum into the cytoplasm, where they'll then act as a second messenger. So these calcium ions can bind to different proteins in their pathway, and they can bind actually to a number of different proteins. Again, this is very important for a coordinated response where many different things need to happen at the same time. Calcium can bind to a number of different substrates to alter their function to produce these cellular responses. So now let's take a look at the responses. So cell signaling is going to lead to either the regulation of transcription or the regulation of other cytoplasmic activities. So the cell's response to an extracellular signal is called the output response. And ultimately, that transduction pathway is going to lead to some kind of regulation of one or more cellular activities. So the response may occur in the nucleus or in the cytoplasm. Many of these signaling pathways regulate the synthesis of enzymes or other proteins, usually by turning genes on or off in the nucleus. So here we have, in this example, a growth factor receptor that's going to go through this whole transduction process, ending up with an active transcription factor that's going to enter the nucleus. That transcription factor is then going to bind to the DNA and either initiate or inhibit transcription to an mRNA, which is then going to be translated into a protein. So this is a very, very common kind of response to a cell signal, the activation of transcription, the regulation of gene expression. So the final activated molecule in the signaling pathway may function as a transcription factor. But there are other kinds of responses that cells can have. So other pathways may regulate the activity of enzymes rather than their synthesis. And so this is going to be a case where the enzyme is going to be already present there in the cytoplasm, just waiting to be activated so that they can be used. And this kind of pathway is really important when a very rapid response is needed. There's no time to go through the whole process of creating mRNA and then synthesizing a protein. We need to act now. In these kinds of pathways, we can just activate those enzymes and they're ready to roll. So the example that you see here is the example that was kind of a thread going through the chapter in the book. Epinephrine leading to the synthesis of glycogen phosphorylase, which is going to convert glycogen into glucose that can be used rapidly for muscular activity. Where here we have the binding of the receptor going through this whole transduction pathway involving adenylyl cyclase, creating cyclic AMP, activating protein kinase A, which is going to activate phosphorylase kinase, which is going to activate the glycogen phosphorylase, which is going to convert those glycogen molecules that are stored in the cell into glucose 1-phosphate that can be used for energy. So this is going to be something that can happen very, very rapidly. So signal could also cause opening or closing of an ion channel in the plasma membrane or a change in cell metabolism as the cellular response. Signaling pathways can also affect the overall behavior of a cell. For example, what we'll see in great detail in the next web lecture, a signal could lead to cell division. As I mentioned before, that these signaling pathways, these responses can be very, very highly regulated to give the very precise response that's needed. So a response to a signal may not simply be on or off, but it could be moderated in different ways depending on the conditions. So remember, these are mechanisms to maintain homeostasis. Conditions need to be maintained within an optimal range, which requires very fine scale regulation. So there are 4 aspects of signal regulation. One is amplification of the signal, and thus the response. So we want to have a big response for a very small amount of signal. We do this through amplification. The second is the specificity of the response. What is the particular cell going to do in response to this particular signal? And we'll talk more about that. And the overall efficiency of the response is another way that signals can be regulated. So this can be enhanced by something called scaffolding proteins, which we'll see in a moment. And also we need to be able to turn that signal off when it's no longer needed. And that's an important component of signal regulation as well. So let's first look at signal amplification. So these enzyme cascades, these multiple step processes, are the main mechanism by which the response is amplified with respect to the number of molecules of signal. So at each step, the number of activated products can be much greater than in the preceding step. So if we think about 1 molecule of epinephrine in this example can lead to the activation of 100, 10 to the second, G protein molecules. So before this unbinds to its receptor, we can have 100 rounds of G protein activation. Before that GTP is dephosphorylated and released, we can have made 10 to the 4th molecules of cyclic AMP. Those 10 to the 4th molecules of cyclic AMP can result in 10 to the 4th active protein kinase. But then these are going to be amplified again into 10 to the 5th active phosphorylase kinase molecules, and then finally another 10-fold increase in the next step to the active glycogen phosphorylase. And then this 10 to the 6th molecules of glycogen phosphorylase can lead to the breakdown of 10 to the 8th molecules of glycogen. So from 1 molecule of epinephrine, this has been increased into a very large response. Again, this is an emergency procedure. This is the fight or flight response. We need to have a very rapid response. This kind of amplification really makes sense in these conditions. So different kinds of cells are going to have different collections of proteins. So even though 2 different kinds of cells may have receptors for the same signal, they're not necessarily going to respond to that signal in the same way. So these different proteins are going to allow cells to detect and respond to different signals. But also the same signal can have different effects in cells with different proteins and different pathways. So for example, in our epinephrine signaling, when it finds a liver cell, it's going to undergo the pathway that we just saw that breaks down glycogen into glucose molecules. But epinephrine is also going to have an effect on respiratory rates. It's going to increase respiratory rate, heart rate, dilation of the pupils. The pupils are going to dilate to let more light in, sweat glands, and so all of these different kinds of cells are responding to the same signal, but they're doing different things in response to it. This is extremely important to that coordinated response. We need to trigger all of these effects at the same time to produce this coordinated response. So some of the ways that this is regulated by this specificity is by something called pathway branching and something called crosstalk. This is going to further help the cell coordinate these incoming signals and regulate them. So what we mean by pathway branching is, for example, maybe in this kind of cell, the signaling molecule just causes one single pathway and one single response. But in other cases, the signaling molecule might end up with an intermediate that can interact with two different kinds of substrates that are going to trigger two different pathways and two different responses. So this is pathway branching, leading to two separate responses in the same cell. Again, important for the coordination of responses when a bunch of different things need to happen at the same time. You can have them respond to the same molecule. So let's take a look at what we mean by crosstalk. So this can get a little bit complicated, because this is really the mechanism that allows a cell to take in input from various different sources and integrate that input into an appropriate response. So this is a situation where separate pathways are going to influence each other in different ways. That creates a highly regulated response. So for example, we've got signaling molecule A that has its own pathway and its own response, but some of the intermediates in this pathway are going to affect other pathways. So, for example, this A1 molecule is going to inhibit the receptor for pathway B. So when A is activated, B becomes inactivated. The receptor won't bind to its signaling molecule. But it may actually stimulate the activity of pathway C, increasing its response. And so we see something similar in pathway C. Pathway C is stimulating pathway B. So this is going to happen when B and C are activated in the absence of A. So this is a way of taking different kinds of input from different pathways. Some of them are saying, go, 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 we need this response. And the other ones are saying, you know, hold off a little bit. We don't quite need so much of this response. And they're going to kind of moderate the effects of one another. And this is called crosstalk. Another way that these pathways are regulated is through signaling efficiency. And one of the ways that this happens is through scaffolding proteins. The idea behind scaffolding proteins is similar to what we were talking about with proteins being embedded in the cell membrane, and that kind of holds them close together in the right order to go through a series of steps, for example, in a metabolic pathway. Scaffolding proteins are proteins that are going to do this. They're going to hold different protein kinases close together. So these are large relay proteins to which other relay proteins can be attached. So you don't have to wait for these things to kind of diffuse and bump into each other. They're held together in the right position to be able to undergo these reactions immediately. So scaffolding proteins can increase the signal transduction efficiency by grouping together different proteins involved in the same pathway, so they're always nearby ready to react. So in some cases, scaffolding proteins may also help activate some of the relay proteins, so it can actually be one of the relay proteins in this pathway. Finally, the signal has to be turned off at some point when that response is no longer needed. Very bad disorders occur when there's no mechanism for turning these things off. So inactivation mechanisms are an essential part of cell signaling. If the concentration of an external signaling molecule falls, then fewer of these receptors are going to be bound to their ligands. So unbound receptors revert to an inactive state. So remember, these ligands are binding reversibly. So they're going to bind for a period of time and then they're going to unbind and diffuse away and become inactivated. These unbound receptors are going to revert to an inactive state, but remember also that all of these activated proteins are going to be reverted to their inactive state through these protein phosphatases that are going to remove those phosphate groups. So finally, we're going to use this idea of programmed cell death to integrate all of these ideas about cell signaling and cell signaling pathways. So cells that are infected, damaged, or at the end of their functional lives often undergo what we call programmed cell death. They're sometimes referred to as organized cell death. So apoptosis is the best understood type of programmed cell death. And in the case of apoptosis, this is an organized form of cell death. So the components of the cell are chopped up and packaged into little vesicles that are then going to be digested by macrophages and a vertebrate system, these scavenger cells that are going to digest the contents. So this is really important because if you just sort of let the membrane rupture and let the cell spew its contents out into the environment, you're going to have a lot of enzymes, particularly digestive enzymes, leaking out of the dying cell, and that's going to have a bad effect on neighboring cells. So we want to do this in a very organized and contained way. And so these little vesicles are known as blebs. So when you see this cell undergoing apoptosis, it's going to have these little vesicles forming all over it. And those little vesicles are then going to be engulfed by phagocytosis by these scavenger cells. So one example of apoptosis occurs during the formation of digits in land-living vertebrates. So all of the hands and feet and paws that you see invertebrates started out during development as these paddle-like structures, completely webbed, with what's called interdigital tissue in between. And as development continues, these cells are resorbed. So they undergo apoptosis, their materials are taken back up by these scavenger cells, and more of this tissue is absorbed until finally you've got the fully separated digits. So this is also the kind of process that happens when tadpoles turn into frogs. They have to get rid of that tail. That tail is resorbed by apoptosis. So let's take a look at how this happens. The example in your book is from the Soil Worm Caenorhabditis elegans. So in worms and other organisms, apoptosis is triggered by signals that activate a cascade of suicide proteins in the cells that have been programmed to die. So in the normal, not marked for death of cells, cells that are destined to live at least for a while, or there's no ligand binding with this receptor for the death signaling molecule. And there's an activated protein called CED9, stands for cell death 9. When this is active, it's going to inhibit the activity of CED4 and CED3. So these are steps in a pathway. So it's directly going to inhibit CED4. And these are proteins in the cell death pathway. So as long as there's no death signal bound to this receptor, this protein stays active and continues to inhibit the death sequence. When the death signal is received, this CED9, this apoptosis inhibiting protein, is going to be inactivated. So it's going to stop inhibiting CED4 and allow it to become active, which is then going to activate CED3, which is known as a caspase protein that's going to promote apoptosis. It's going to activate things like nucleases, it's going to break down nucleic acids, and proteases that are going to break down proteins. And here is a cell forming blebs, so these vesicles are surrounding the discarded products after these nucleases and proteases have been out them. So the chief caspase in the nematode is called CED3. That's going to activate these catabolic proteins that are going to break down the macromolecules. So we see in this example all of the things that we've gone through, this idea of a receptor. So that can either be a G protein coupled receptor or an RTK, receptor tyrosine kinase, that's going to activate some kind of second messenger, that's going to go through some sort of cascade, probably a phosphorylation cascade, possibly with amplification of the signal along the way to create a response, which will be the shutting off, the inactivation of this inhibitor protein that can then go through the rest of this pathway toward apoptosis. So this is the basics of cell signaling. In the next web lecture, we're going to see one of these cases of cell signaling in the form of growth factors, which are going to be a key component of regulating the cell cycle. So the cycle of replication of cells for either growth or reproduction.